Okay, we're back. And what I wanted to do is to talk to you guys about this balance of CO2 and water and how it's converted by carbonic anhydrase into carbonic acid, and then ultimately becomes a proton and a bicarbonate. And this movement follows Le Chatelier's principle in that we have these different reactants here that then end up producing products on this side. But the whole process can go the other direction, as we learned in the respiratory part of pulmonary physiology, where we can have an excess amount of protons, for example, or bicarbonate, that would then push the reaction in the opposite direction. And so this interaction between CO2, water, and then how it can actually be converted into protons and bicarbonate, or how protons and bicarbonate can be converted to CO2 and water, is what the body uses to help take advantage of being able to help regulate and get rid of metabolic waste. As you may recall, CO2 is one of the things that actually is a byproduct of metabolism, and protons are as well. And so how is it that the body actually gets rid of it? And a key sort of underlying principle that you can kind of think about is that CO2 is removed by the lungs, and so that's how we're actually able to exhale and get rid of it. Whereas protons and bicarbonate, you can't remove it by the lungs. The only way that you can help to get it out of the body is for the renal system to excrete it. You got to pee it off. So that's how we manage it. So in the renal physiology component, what we're emphasizing here is how we have two different types of cells, type A cells and type B cells, that help us to be able to do this job of being able to excrete either protons or bicarbonates respectively. So this connects to slides that we have in our PowerPoints. And what I've done is I've zoomed in on slide 19 that we have to show what we have for type A cells here. So type A cells, what this diagram is showing for us is the lumen of the collecting duct. It's helpfully colored yellow, so it's sort of pre-urine, if you want to think of it that way, on its way out. And then what you have is your type A cells that are intercalated into the nephron. And their job is essentially to help be able to pump the protons out. So some of the details matter here, so we have to kind of keep track of it. We have our lumen of the collecting duct here that has some potassium that's been filtered through it and so forth. That's just been presented as it normally would. And then what we want to think about as we're looking at this diagram is that we have carbonic anhydrase stacked up inside of these cells. And this helps us to be able to take the CO2 and the water and convert it into protons. And the protons then can be pumped out. So the CO2 is coming from the blood, for example, and diffuses across and then is able to actually be able to be excreted out through these proton pumps that we have in the type A cells. We also have a transporter that causes reabsorption of potassium, so that as we pump out the protons here, we take in the potassium here and we reabsorb it. So this helps to elevate our potassium inside of our bodies. So what can happen sometimes is that if we have an excessive amount of CO2 and protons that we're trying to get rid of, then what will happen is you can end up having an increase in the amount of potassium that ends up in our blood. So let's walk through that just to make sure we're clear on it. Here we have protons that are really high, meaning that your pH is what? Is it really high or really low? That means that your pH would be really low. If it's very acidic, then your pH is low. So you have lots of protons here, meaning your pH is low, it's very acidic in the blood, for example. And what it's going to do is that it's going to bind with bicarbonate and then convert it into carbon dioxide. And that carbon dioxide can then, in turn, be able to cross into these cells. And once it gets inside the cells, it can bind with water and, through the action of carbonic anhydrase, be able to be converted back into protons that can be pumped out. So why go through all this trouble? Why not just have the protons go into here and pump it out? And the reason is because we have cell membranes that make it difficult for the protons to actually escape. And so it's easier to convert it into a gas that can diffuse into the cells and then be able to be converted into a proton that can be pumped out. So it's the way that our renal system handles these protons is by working with carbonic anhydrase and converting carbon dioxide into these protons that can be released. Okay, so what about the bicarbonate that's actually made out of this whole mess? So we have we're separating and we're sorting out, we're putting out the protons here. The bicarbonate is actually taken back into the body and reabsorbed into the body, which then goes into our blood. And that's useful because the bicarbonate can then bind to protons that are present in our blood and be able to help keep this cycle going. So we save our bicarbonate here so that we can actually be able to use it to capture more of these protons that we have in excess in our blood. So all of this type A cells that we're talking about here is the action of it is predicated on the idea that you have lots of protons or you have a lot of acidity in the blood, a low pH. So let's go back then to what we talked about in what we have here. In this equation, what we've talked about is we have these protons and we have type A cells is what we've been focusing on. And this is all about having the excretion of protons into the nephron from the blood. This is an ATP-dependent process. And we talked about how we have two different ATP transporters, one that's just a proton pump, and the other one that's an antiporter, right? The one that's pumping out the protons out into the nephron and bringing potassium in. And that this is an acidosis, and this is active during acidosis. And so that's a useful mnemonic for thinking about what type A cells do, is that it begins with the letter A for acidosis here. And the proton pumps are on the apical side. So just so that you can see what I mean by apical, the apical side of this cell is the one that's facing the nephron, the interior of the cell. And the basolateral side is where the cell is actually anchored in the rest of the body. So when we think about how cells are oriented, if you can remember from 208, the main idea is that you have some kind of, where the cell actually has a base, and then you have a part where it's actually on the apical side where it's reaching into somewhere else, and as is the case in these cells. So, fine. We have this kind of, we have the placement, and we're going to remember that term apical as a term of orientation there. We're remembering what the pumps do and how they're able to get protons out. What about bicarbonate? These type B cells are active when we have when our blood is basic, and so you can remember that term by B for basic for that. And it's all about reabsorbing the protons and excreting our buffer, our bicarbonate buffer. And so really it's almost a mirror image of what we see in the type A cells. So let's take a look. Here's our type B cells. And if you remember, our type A cells had these two pumps located on the apical side, but now they're on the basolateral side. And the bicarbonate transporter that we talked about here is on the apical side. So really you just got to remember who these different players are. And then if you can remember which side they are in one case, you can just do the mirror image for the other. And it ends up making sense because you want to be able to keep in those protons and be able to get rid of that bicarbonate so that you can help elevate the concentration of protons in the blood and make it so your blood is no longer so basic. And if you remember, you're trying to, so the situation that happens is if your blood is basic, that means your pH is high, and you want to bring it down by keeping some of your protons. Okay, so in terms of acid-base disturbances, what we are looking at here is a number of different cases here. We have respiratory acidosis, metabolic acidosis, respiratory alkalosis, and metabolic alkalosis. And there's a lot of different components of this. I'm going to talk about the acidosis ones the most. And what we're thinking about in this is on our sheet, what we see is that in the case of respiratory acidosis, what I want you to just basically imagine is in respiratory acidosis, our primary problem here is we have some kind of deficit in our pulmonary function. So I just wrote down lose our lungs somehow. And so here we have CO2, I circled it, and what's going to happen is if we lose our lungs, we have a hard time removing CO2. And so we're going to have this kind of buildup of it. And so what I would have asked you guys in class is I would have said, so what's going to happen to the concentration of protons in that situation? And here, though I've already given away the answer by showing you that if you elevate your CO2 levels, then what you find is that you have an elevation in the concentration of protons. Your pH would drop. And so your kidneys are going to work to compensate for this. They're going to, and so they call this renal compensation. Your A cells, for example, would kick in, and they would excrete the protons from the nephron into the nephron from the blood. So that's the basic function that we would be seeing in respiratory acidosis. In the case of metabolic acidosis, the issue is different. The source of the protons is by different types of things. One thing it could be is from a damage to the kidneys. It could also be from a source of extreme exercise or some other kinds of conditions which you may be generating a lot of protons or acids, and then that in turn would elevate this. And so in class I would have said, so now what would happen to CO2 levels? But I'm giving it away here by showing that your levels of CO2 would actually increase in the blood because you would have had more protons. Why? Because it's this action of this equation. More protons over here would ultimately get converted into CO2 and water, and then we breathe it off. And so what you have in the case of metabolic acidosis is that because you're not able to get rid of the protons very effectively, or you can't keep up with it very well, then you're going to generate CO2. And so these patients will have a faster rate of breathing to try to help increase alveolar ventilation and get rid of their the CO2 that's in their blood. So those are two types of compensation that we see. The other types that we have here, respiratory alkaloidosis, this can happen sometimes in the case of hyperventilation, in which case we've actually breathed off so much CO2 that we have a deficit of protons that are present. And metabolic alkaloidosis. So this can happen when you lose a lot of protons from vomiting. So if you vomit a lot of stomach acids can be lost, and that can make it so you end up having a more basic kind of environment, or hypoventilation, and so on. So, the hypoventilation actually, in this case, would be a way to compensate for this metabolic alkaloidosis. And that part we can think about and appreciate as we look here. So if we have a deficit of protons here, and we don't have enough of them, then what we can do is that we can accumulate CO2 by letting our lungs work less. We remove less of the CO2, and we're able to generate more of that, so it's able to push things along towards the other side and help us get more of our protons. And so that's what that note of hypoventilation is referring to. All right, so I hope that's helpful for navigating and thinking about some problems that we have related to this. Focus on those different types of protons, where they're located, where the pumps are, and where they're located. Think about what happens when we have respiratory or metabolic acidosis. There's some problems in the exam review sheets also that are helpful in terms of thinking about this as well. And that's it for this component.